This is actually a presentation I gave on a conference on silicon nanotechnology with biomolecular sensing, not in general, but what we do in our group then. And there are small pictures down here. So this is the ion field effect transistor, which I will start with. Here is a nanopore, which we're trying to develop in our group. And here is a microcapillary, and that part is what we're using quite a lot right now, and which Urba has developed, and he will show that. So I have an introduction which you, which you have partly covered now. Right. Yes, so I mean, many years ago we were sort of, the microtechnology was progressing very well, and you could assemble a lot of transistors. And nowadays, at the university level, we cannot really make state-of-the-art devices like they do at companies when they develop your mobile phone chips, for instance. They're extremely complicated, of course. So we're starting to giving up that, but we have looked at, we have a very good clean room with both silicon process technology as well as for 3.5s. So we looked into other type of applications, and indeed biosensing and is one of the applications like that. So this is, as you've probably seen, this development in transistor technology, which is named by Moore's law, which seems to be like a natural constant here. Development, as you know, the technology is advancing like a factor of 2 number of transistors every second year or something like that, which seems to go on forever, but we know that there is a limit. So the concept was then add-on functionality. More than more was named, and there are lots of such European calls, I guess, for technology. And one of the things that people have in mind is that maybe in medical technology, so you sample a patient here, you get some sample from your blood. It's sent to a lab, and you get a lot of different things out of that, characterizing your body. And maybe all of that lab can be scaled down to some kind of chip like this. So you would have transistors on the chip, but you also add on some kind of microfluidics on it so that you can introduce samples. And yes, what you talked about, the DNA sequencing is of course a big thing right now. And as you know, the first genome sequencing was taking like 10 years to complete. And now we're down to, I think, roughly 12 hours or something like that for the whole genome sequence. And we have such machines at Royal Institute of Technology. It's not where we are, it's a separate campus which is close to the medical school. Yes, this is what Urba showed before also. And remarkably, this the cost per genome, so it was tremendous when it all started, of course, but it's been doing like that, but it's going much faster than Moore's law, in fact. So now we're down to, it was named like the $1,000 genome, and we are there at the moment. And I think you can also send in your sample and get your genome analyzed for some cost. $1,000.
No, companies are there who offer free service, but you have to agree that they can use this data for their business purpose.
For example, cloning.
Yeah. but then you have like the $1 million interpretation of your genome, if you want to know what it really means. Yeah. So yeah, this is Professor Joachim Landerberg. He has a tool like that. Interesting. They were doing actually the genome of the spruce, or what is it called, spruce? The trees, you know, what you can make Christmas trees from, but a big one. And that genome is 10 times as big as the human genome. So we're not the most clever, maybe. But it is due to that the genome also for the spruce has developed over a much longer period than for mankind. Yes. So why is it? Why would this be good? Well, with CMOS technology, you could possibly do multiplexing. So for instance, for certain cancers, you can get like the what's called for men this PSA cancer. You can send in and they probe one protein, for instance. But usually biology is much more complex. You would like to probe many different proteins or so. And that's something maybe one can do on a silicon chip. Probably it can be done so that you can have a very fast analysis. Maybe you can get the answer in a couple of hours or less. It could be cheap, and if it's really cheap, it could be disposable. So, I mean, if you look at regular chips, they can be maybe a tenth of a euro per chip or something like that in large fabrication scales. Yes, the last thing also, yes, miniaturization. Maybe you can also eventually, maybe you can send in chips into your bloodstream and get some radio information back or something like that. Yes, so, and I had a student we used EV lithography, and this was probably around year 2000 or 2005 maybe, so yeah, when he started. And he suggested that, why don't we make nanowires? Because at that time there was a method developed where you could grow nanowires vertically. It was called the VLS, vapor liquid solid method, yes. So it's in a petaxis system you can have it's called catalyzing particle, so that you could grow vertical nanowires like that. And we had a big clean room with good silicon technology, and we could also use EV lithography. So we said, why don't we make this in pure silicon technology instead? Because in this way, when they make them nanowires, they had to cut them and sort of dispense them onto a surface which had electrodes. And of course that's a very random process, so we thought that why not try to make it in pure silicon technology. Anyway, the results were that they showed then by this technology with the grown nanowires that they used on electrodes were sort of fantastic. So here's a measurement that a group at Harvard, Charles Lieber, showed, and they were using then, they had some kind of probe DNA onto the nanowire, and then they introduced 60 femtomolars. So femtomolar is 10 to minus 15 molars. So a very small fraction of molecules, and they got a very nice response here. Here's another measurement from another group at Yale University, published in Nature, of course. And they have a sensitivity of 10 femtomolar, and look at it's very fast response here. Here's a Chinese group showing similar things. 0.1 femtomolar, fantastic. And yes, I probably already talked about this technology. So In fact, it works almost like an FET, so you drive a current through the nanowire, and if you have some change in charge, which could happen when proteins, what do you say, like recombine? No, they protein. Yes, when they come to antibodies on the surface, you get the charge change, and therefore you get the change in the current here. So we started to do that, and this is from our samples. So we made by e-beam lithography very small nanowires, and those are typically made on silicon on insulated wafers. You can buy those. It's regular silicon wafers. They have an oxide, and on top of that oxide they have another silicon layer, which is the device layer. So that's very much used, and we etch down the device layer so it would only be like 30, 40 nanometers. Later on, we realized that we can make these nanostructures not by making e-beam lithography, but if we make the thickness very small here. So typically we etch down the silicon to something like 20 nanometers only, and then we get the sensitivity really to, and then that's easiest that we can use ordinary photolithography to make this. And this is a schematic then showing that this is our device which we make, and we put the electrodes here, and we're driving a current here, and the hope is then that we'll bind some streptavidin in that case to the biotin, and we can see a charge change, and therefore a change in the current here. So this is our measurements then, and you see here we had a very simple device, no microfluidics at that time. This was Niklas Elfström, was the PhD student. So here is for, I can see now this is probably like nanomolar, and the best we could do was to sense picomolar, about 1 picomole here. So that's this point. Not femtomolar, could not achieve that, but we were around picomolar. But now you see here that the response here is quite long here. It's like 1,000 seconds. It's very different from what they showed before. And yes, so so this was the student. He's dressed up here. So this is our results. Now if we compare to this group, we actually talked to this group because we are friends with them at Yale University. And I asked them about how could you get such an instantaneous response here just without infusing. And he told me that, yes, sir, we're stirring very frequently like that. So I told my PhD student, why don't you stir? And he went down to the lab and he was stirring like hell. Still we got the same response here. And then a year later or so, there was a theoretical paper published, and they show that if you have a solution of 1 femtomolar and you have a nanowire like that, for the first molecule, by simple diffusion in this liquid, to find that nanowire, it takes about 1 hour. So to have some response, they probably must have something like hundreds or also. So this is theoretically impossible, and so these measurements have been very much questioned later on. Everybody realized that it's not possible like that. And what is the explanation, you think? Well, if you think of an experiment like this, either you have microfluidic or you're simply dipping into your solution like that. So what you do is you start with your buffer solution, PBS, which is a highly ionic solution. and then you insert the other another PBS where you have your analyte molecules, what you want to detect. And imagine that there's just a slight difference in pH or something. the sensor will immediately sense that. Or if there is a change in concentration of that. So I mean, just practically, it's very hard to do that experiment. So this is probably the explanation why they get instantaneous response here. It's never been shown or disproved or anything like that. While this is what you normally see for a biosensor, that there is a rise here in your signal which depends on time like that, and the derivative here indicates your concentration of your analyte solution. Another problem you have is that there is device screening. So any electrical sensing is very difficult because you have a very ionic liquid, and any charge then will be screened by these charges. And effectively, this is a schematic of a protein, it may be, the charge distance may be only 0.1 nanometer or so. So what we typically do is we lower the concentration of the PPS 10 times. You cannot lower it to very low, because then proteins and DNA don't like that solution. So they will be deformed or so. Another problem you have with this kind of sensing is that although a lot happens, you want to detect what happens here, in all experiments you need a counter electrode here. And there can be also some chemical reactions at the counter electrode when you introduce the molecules you want to sense. Yes, so Apurva was showing how we make our chips. So this is a final chip. This is a 4-inch wafer with lots of chips. And we start by defining the silicon nanoribbons. So we have this top layer, device layer, and we make lithography to form the nanoribbons. We call them, instead of nanowires, we call them nanoribbons because they're flat like that. Then we put metal contacts, so on top of that. So you see that structure. And finally, we put an insulation and microfluidics layer on top of that. And the microfluidics layer you see here. And here's a chip ready to be measured. Yeah, this is the layout, and here are we have some different shapes of these nanoribbons here. One is a very long one. And these are typical characteristics of these devices, ID characteristics. And yes, Purba already touched upon it. One can use this plotter then to functionalize nanowires. And the reason why we bought this is actually we want to have lots of nanowires, and they will have different antibodies, so we can do multiple sensing. One of the things we discovered then was that if we had functionalized different, the same functionalization on nanowires here, we saw that there were different signals from each of them. So the different nanowires here show different amplitude of the signal. And we couldn't figure out what that was. Similarly, if we functionalize just one of the nanowires here, the rest of them also responded. And Apurva was set to look into that, and finally that came out as a new technology.